So first, there are also guidelines that are very recent. So one from January 2020 from FDA, where you can read a lot of the CMC details about it. There's also one from EMA with a similar title from 2018. So not to go through all these documents, just an example on the impurity chapter from the FDA guideline. We recommend that your manufacturing process be designed to remove process and product-related impurities, and that you have tests in place to measure the level of these impurities. And then at the bottom, list different types of impurities, including viral proteins and reagents containing other proteins. So clearly there are some good guidelines how to do this, but it doesn't say exactly what methods to use, because these methods are not always present or often not in place. So why is it so difficult to measure these type of products? One of the reasons is that they are very complex. The samples are very complex. It's a mixture of different proteins from different sources. I've listed some of the sources up here. So one is the viral vectors used. So each of them contain a number of different proteins that are part of the product. It also has a production and packing cell line. So typically insect cells, ASF9 cells, different types of human cell lines like the HEC293 cells, but there are also more rare cell lines used by some customers, for example, murine cell lines. And then we have the manufacturing system and the growth medium and reagents used during the manufacturing. So typically we see bovine serum used in the growth medium, human albumin, antibody columns, enzymes added during the manufacturing. That also complicates the samples. So it is a mixture of many different proteins and many different components that can influence with the analysis. So this makes these complex products very difficult to measure with classical protein analysis methods and classical impurity assays. For example, ELISA is only developed, as you know, for one species of protein. It cannot measure all across different species.